Welcome everyone to the final day of OCB 2021. It's been quite an adventure with all of you. This is our first ever virtual summer workshop, and thanks to e-poster boards, it was about as good as it could possibly be for a virtual meeting. My name's Heather Benway. For those who don't know me, I'm a senior research specialist at the Woods Hole Oceanographic Institution and the executive officer of the Ocean Carbon and Biogeochemistry, or OCB project office. I'm an oceanographer with interests and background in paleoceanography and marine biogeochemistry. OCB is a bottom-up program. I've said this a million times, but I can't say it enough. That means that the leadership and the activities of OCB are shaped by its community through an open process. I just want to, again, every day, be grateful that this program still exists. Thanks to our sponsors and our physical host. OCB was founded in 2006. And since then, for, 15 years now, we've been receiving sustained funding from NSF and NASA, and we've been hosted by the Woods Hole Oceanographic Institution. I also want to thank our event hosts, e-poster boards, especially big shout out to Mike and Chase and Catherine and Eduardo, and it's so many wonderful people that work for e-poster boards that have set up this great space for us. I want to thank our scientific steering committee who put together these amazing sessions, and our session chairs. Usually we try to make sure that our sessions are chaired by an OCB member and a non-OCB member so that there's a partnership there. And these sessions were just knocked out of the park this year. I want to thank all of the speakers who put so much time into putting nice talks together and pre-recording them and trying to add closed captioning so that they could be accessible as accessible as possible. I want to thank my team. Teamwork makes the dream work, and working with May Mahegan and Mary Zawoski is an absolute pleasure and a privilege every day. So can't say enough about that. And I want to thank you for being here, because having you here is what makes the meeting stimulating and fun. So thank you so much. This workshop was planned while working on the ancestral lands of the Wampanoag Nation, made up of the Mashpee, Aquinnah, and Herring Pond tribes. We honor and respect the many diverse indigenous peoples still connected to this land and the lands of our participants. We acknowledge the painful history of genocide and the substantial traditional and local knowledge that indigenous people hold and sustain. I want to point you to the orange Code of Conduct and Incident Reporting button. If you experience or witness any harassment, bullying, or intimidation, discrimination of any kind, please click on the orange button. There's an anonymous reporting form in there, and I really encourage you to use it so that we can address it immediately. Our virtual meeting space is pretty limited today. You came into the lobby, that's your entry point every day. And we're also going to have a happy hour there, a farewell happy hour at the end of the day. So that's the lobby. All of the other meeting areas, except for the lecture hall, are grayed out here because we're not using them today. We're using the lecture hall for all of the exciting new OCB activity report outs that are happening. I do want to say one thing about the posters. If you missed the live poster presentations, the poster hall, the virtual poster gallery, you can see that button over here on the right side, will remain open through the end of the summer. So you can visit any poster presentation you would like. They all have an MP3 recording that goes with them, so you can get the run through on any poster, and then you can reach out to the author if you have questions. The poster directory also has a full list of all of the posters that were presented. The workshop website will include all of the recordings from the meeting, links to the recordings of all of our plenary sessions together, and today's agenda is a quick pop-up here. You can just click on it and a little pop-up will show you what's happening at what time today. Again, if this is your first time joining, take 5 minutes during a break to watch the welcome video, and this really helps orient you to the platform quickly, but it should be pretty easy today because we're just in the lecture hall. We're not moving around too much. So the lineup today, first we're going to have a presentation by Adam Martiny and co-PIs, excuse me, Nicole Poulton and Harriet Alexander are joining us today. Unfortunately, the other two co-PIs, Jason Graff and Sophie Clayton, have other obligations and can't be with us, but they have contributed an enormous amount to this. So you'll hear a presentation from Adam, and then we'll field some Q&A from you, the audience. Next, we'll have an activity report out on coastal Arctic biogeochemical data synthesis in preparation for the NASA Arctic Colors field campaign. And this will be given at 1 o'clock. At 1:30, we'll have another new OCB activity report out on carbon isotopes in the ocean, ensuring high quality results in the future. Finally, we'll have some closing remarks and some fun polls and giveaways of OCB swag. There's some pretty fabulous OCB swag like water bottles and utensil kits, tattoos for your kids, because every kid should have the biological pump and floats and gliders on their arm this summer. Finally, we'll wrap it up with a farewell happy hour in the lobby, and the lobby will be closing today at 3:30 p.m. So you can go out and have a happy hour on your deck if you like. I would like to see how many people we have in the room. Mike, could you maybe tell me that? Is that easy to come on and tell me?
Yes, 31 at the moment. You can always see it in the upper left corner as well.
Oh, thank you. All right, so what I would like for you to do is go to www.menti.com, please, and enter this code. I'm going to add that to the chat as well. Or maybe Mike or May could add that to the chat for me. I have so many things open. There we go. Thank you, May. All right. Now bear with me. I'm going to stop sharing this window. Does everybody have the code? 26610681. I'm going to stop sharing. I'm going to share a different window. Oh, wow. People are putting it in already. This is great. Describe OCB in one word. We've got a little word cloud coming through here. Lobster. Oh, this is fantastic. Synergy, groundbreaking, dynamic, inspiring. I love it. All right, I'm going to advance to the next slide. So get your last answers in there. What was your favorite part of OCB 2021? Thematic plenary sessions. Still leading the pack. Somebody gave the posters some love. One person, 2 people gave the posters some love. 4 people. Do I hear 5? No, just kidding. This is great. Thank you so much. As usual, and this happens every year, it's the plenary sessions, because they really cut across so many of OCB's scientific focus areas, and they really are a big draw. And our scientific steering committee and the session chairs that work with us just really put so much thought and effort into putting the topics together and finding the right speakers. So again, thank you so much. All right, I'm going to sharing now. And then I think I am ready to hand it off to Mike to give a little brief how do you use e-poster boards?
Yes, it'll be very short today. So hi everyone, welcome. And we've loved to be your hosts in this virtual conference software. Thank you very much for joining us. As we've said a couple times before, there is a little bit of a learning curve with this software compared to a typical Zoom or Teams meeting. So we want you to know that you can find us for tech help, and we like to ask for patience and grace as everyone learns how to navigate the software. Some presenters and some attendees, I believe, are still here, or I should say are here for the first time today. So right now we're in stage presentation mode, and we're in the lecture hall event space. The stage presentation takes over everyone's screen, so we all see what's on stage regardless of what table we're at in the room. If you're not a speaker, you can interact via the chat or the Q&A panel at the right side of your screen. You can enter things in the Q&A anonymously if you choose. You can also click items in the Q&A to upvote questions or comments that are there if you'd like to see them featured or addressed by the speakers during the Q&A. There will be several Q&A periods today. Throughout the event, in all active spaces, so that today again includes the lecture hall where we are now and in the lobby, you'll be able to find e-poster board staff if you need help. If you connect to any space labeled help desk, we should be there to help, and you can also double click and join us wherever we are on the map. You can also message us via the participants list or the chat when we're in stage mode as we are now. So one last comment, which is very important. Refreshing the web page, this web page in your browser window will resolve most of the technical hiccups that could happen with the software. For example, if you suddenly can't see or hear the stage presentation, try a refresh or even multiple refreshes on your browser window, and you'll likely find that that resolves the issue. So with that, we're going to bring up the speakers for our session. Thanks for your attention, and we hope you enjoy the day.
Hi, everyone. Can you hear me? Okay. Maybe you can't answer. All right. Today, I would like to present this new project that we're trying to establish, which is called Biogoship. It is basically the idea to integrate the biological measurements on a large number of sectional surveys. And the work has been so far supported by both NSF, NASA, and NOAA, and we work closely with Ghostship. And if you're interested in knowing more about Biogoship after this presentation, then I encourage you to go to biogoship.org to get more information. Also, the vision for today is really to have an interactive session with the OCB community. And so I strongly encourage you to post questions in the Q&A, either during this presentation or afterwards. But just feel free to post during the presentation. And we'll be very happy to answer them either then, if it's directly related to some details, or afterwards. All right, next slide. So the vision of Biogoship is really to develop a deep understanding between the physicochemical environment and global ocean plankton biodiversity, abundance, and carbon stock, as well as the biogeochemical role in the context of a changing ocean. And a lot of this has come about as an idea to leverage this already large investment that both the US, but also the international community has been put into the Ghostship platform with all the different sectional surveys and its complementary high quality hydrographic measurements. Next slide. So, so far we've assembled a really strong team, but this is a community effort, and we're certainly very interested in people here and the wider community to get involved. So beyond myself, it's Harriet Alexander from WHOI, Sophie Clayton from Old Dominion University, Jason Graff from Oregon State, and Nicole Poulton from Bigelow. We're sort of the primary investigators in Biogoship. But I would also like to highlight Heather Benway and OCB has been a very strong supporter from the beginning. Also, Emmanuel Barth has been our cheerleader and instigator from day one. And more recently, Luke Thompson from NOAA has started to become quite involved, and we're developing a deep partnership with NOAA I'd also like to acknowledge many past members of my lab who were sort of instrumental in getting this started, as well as the Ghostship community, who's been extremely supportive of this idea of integrating biological measurements. Next slide. So before I talk about Biogoship, I felt that it would be relevant also to just sort of briefly summarize Ghostship. I imagine that many of you, at least some of you, are familiar with Ghostship, but it may also be new to some of you. Ghostship is sort of the most recent iteration of these sectional surveys that started with the World Ocean Circulation Experiment, or WHOI. And the idea of Ghostship is that it's an international ship-based global survey of ocean hydrographic sections. The idea is that every 10 years or so, with shipboard operations, most, if not all, ocean regions are being covered. And then after a decade, then you go back and then you sample again. And so that through a decade, we can get a deep understanding of, with Ghostship, of the physical and chemical environment in the ocean. But by then repeating these surveys every decade, then we can also monitor the ocean as it's changing, for example, due to both natural but also anthropogenic forcing. Another part of the vision behind Ghostship is the idea of standardized measurements. And so Ghostship has spent a lot of effort into calibration to ensure that we can compare the measurements through time and space. And some of the things that has come out of Ghostship is a strong understanding of the link between surface and deep ocean processes as the sampling is through the full water column. In the current version of Ghostship, there's a strong focus on the physical and chemical environment. And some of the things that we've learned through the Ghostship effort and science is, for example, a quantification of the uptake of heat in the ocean, as well as anthropogenic carbon. There's also a lot of measurements that are focused on nutrient cycling. So a lot of our understanding of the variability as well as drivers of nutrient cycling is based on sort of these high quality measurements from Ghostship. More recently, there's also an increased emphasis on oxygen, and particularly the observations that there's a decline of oxygen in the ocean, deoxygenation. And a lot of that is based on Ghostship measurements. Also, more recently, Ghostship has been quite involved in taking high quality measurements simultaneously with the deployment of autonomous floats. It's been used for calibration of, for example, the Argo fleet. Next slide. So here's a map of the different sections that we have already participated in red, and in yellow, it's 3 new cruises that were just supported by NOAA and NASA. And then in the black is proposed sections within the next 5 years. And so as you can already see from this map, is that there's an incredible global coverage of these shipboard sectional surveys. And so by participating and starting to take biological measurements, we can very quickly leverage this enormous investment that is already put into Ghostship and very quickly get a very large scale understanding of biological biodiversity as well as functions, biological functions in the global ocean. Next slide. So the idea in this group is that we want to take a suite of measurements along with these physical and chemical measurements that are already being done in the Ghostship. And the measurements that we're proposing is metabarcoding, both of prokaryotes, but also of, and particularly, I would say, of eukaryotes, all the way up to larger organisms. There's metagenomics, and I'll show a little bit of some preliminary data from the metagenomics effort. We're also proposing to do more metatranscriptomics as we go into the next phase of Biogoship as eDNA, sort of similar to the metabarcoding. In addition to all these omics measurements, we're also implementing a variety of optical measurements focused on both fluorescence and spectral absorption and backscattering. And as we have seen in some of the earlier sessions in OCB, right, one of the, for example, Ivana Sedmit's talk on Tuesday, right, one of the powers of adding these optical measurements to some of the chemical measurements is that then we can really fill in and get a deeper understanding, a higher resolution understanding, both in time and space, of, for example, carbon stocks and photophysiology. Another thing that we want that we're planning to do is a lot of imaging and flow cytometry. Here we're planning to use hyperspectral flow cytometry and hope to do that inline so that we can get a lot of physiological data. We're also planning to use the IFCB to get slightly larger size fractions, the UVP, and also the list, so that we can get a full particle size distribution from the smallest particle up to at least in the hundreds of micron size particles. Another set of measurements that we're implementing is elemental analysis of a particular organic matter, so both carbon, nitrogen, and phosphorus, as well as getting a sense of how much oxygen it takes to respire the organic matter that is present in the ocean. Together with NASA, we're also collecting samples for HPLC pigments. And then finally, we're hoping to add acoustics to get a better sense of the distribution of larger organisms. One of the key aspects of this is that by taking this a broader suite of measurements, there's a lot of possibilities of integration and synergies between these different measurements, both by linking omics to various types of carbon stocks and particle sizes, but also with the opticals, as I said, to get a much higher spatial and temporal resolution. But one of the keys in Biogoship is that we are utilizing established techniques so that we can get a scale up to this large global effort. And also enabling the involvement of other partners, for example, the international Ghostship community. And so by using established techniques, this integration between institutions will become much more simple. Next slide. So one of the visions of Biogoship is that there will be a tight integration in scientific objectives between what we can think of the core Ghostship and Biogoship, and also a tight integration between the data that they are collecting and the data that will come out of Biogoship. And I think sort of intellectually, it's probably intuitive that by having a lot of the hydrographic measurements that are coming out of the core Ghostship, we can use that as a detailed characterization of the ocean environment in the full water column, and that can help us inform what are the drivers of, for example, of plankton biodiversity in a very detailed sense. Another aspect that the core Ghostship can provide is an understanding of the physical mixing of the environment, and so we can start to develop a better understanding of the role of mixing versus, for example, selection of plankton in setting the community structure. Next slide. Here's just sort of one example of sort of how substance and integration can look like. On the left, we have a model prediction of plankton biodiversity. This is some years old now, studied by Andrew Barton when he was in McFarlane's lab. And on the right-hand side, we have or just from one transect throughout the Atlantic Ocean, from about 50 north to 50 south, where we also have the biodiversity pattern. And I think if you squint, we can start to see that there are some quite strong similarities in the biodiversity patterns that are being predicted based on some fairly simple ecological rules and some of the biodiversity patterns that we are observing through Biogoship. And so we definitely expect to really expand on these types of analysis when we're getting these thousands of samples from around the globe describing the biodiversity of the planting communities, and really being able to integrate that with model predictions and see if we can understand what is the relative role of environmental selection versus dispersal and mixing. Next slide. What I would like to spend a little bit of time here is that we also envision that there will be a lot of synergies in the other direction, whereby some of the new biological data that we are collecting through Biogoship can really have an impact on sort of some of the questions that have been of interest for a long time in the biogeochemical community and the Nicole Ghostship community. And for example, one of the things that we will provide is a detailed understanding of both of the regulation and the empirical pattern of the carbon to nitrogen to oxygen ratio in organic matter produced by biology. Next slide, please. So one paper that came out recently is related to the relationship between the carbon and the oxygen cycle. So the relationship between the carbon and oxygen cycle via the biology comes back to the Redfield ratio, now proposed nearly 80 years ago. And if we look at the top, we have basically the chemical equation for the respiration of organic matter proposed by Alfred Redfield. And there we can see that it takes 106 moles of oxygen to respire organic matter with the chemical composition that he proposed. And so with these numbers in mind, we basically get a relative ratio of oxygen to produced CO2 of 106 over 106 equal to 1. If we also include the oxygen demand via nitrification, we get another 32 moles of oxygen that's required to oxidize the ammonium into nitrate. And then we get basically the overall quotient between oxygen and carbon to mineralization to nitrate of 1.34. And these numbers are really integral in many biogeochemical concepts, both in models, but also other places. Next slide. So this is data from one of the Ghostship cruises from near my home in California and all the way down to the ice edge in the Eastern Pacific. And what we see is that the amount of oxygen that it requires to respire molar carbon is considerably higher in subtropical and tropical environments and decline as we approach into the Southern Ocean. And so it's near the Redfield proportion in the Southern Ocean, but considerably higher in low nutrient warm regions. And we don't really know why that is, and we can discuss that later, some of the biological mechanisms. But I think for the sake of this presentation, the key is that it's not constant. So basically, it requires a variable amount of oxygen to respire molar carbon. Next slide. So, for example, with the deployment of all the BGC Argo floats, one of the key things that in biogeochemistry that this has been used for is to use the drawdown of oxygen and nitrate to estimate net community production and carbon export. However, in all of those calculations, there's a requirement of the elemental conversion from oxygen to carbon or from nitrate to carbon. And so therefore, having a detailed understanding of the C to N to P and minus oxygen ratios could be quite important to reduce the uncertainty in these calculations. Also, many inverse models and, for that sake, forward models uses hydrographic data to estimate biogeochemical cycles and often involves these elemental conversions. Another place is in the uptake of anthropogenic carbon, where also these elemental ratios play a role in these calculations. Another key objective of the called Ghostship. And again, having good measurements of the elemental composition of these particles in the surface and at depth will greatly help with understanding the linkages between the nutrient cycles, oxygen cycle, and the carbon cycle. Next slide. Another place where you can envision integration is via remote sensing products. Next slide. So some of you may recall that Elise Larkin gave a presentation in the first day of the OCB workshop, where she worked on using metagenomic biomarkers to estimate nutrient stress. This data was primarily coming from the participation in Biogoship and showed a clear biogeography of nitrogen, iron, and phosphorus stress, as you can see on this image. Next slide. We're now working with the remote sensing community to see if we can link the distribution of these biomarkers to remote sensing of, for example, chlorophyll and carbon. So many laboratory experiments have shown that the chlorophyll to carbon ratio is quite sensitive to nutrient stress, whereby when cells are nutrient stressed, they are frugal on the production of chlorophyll. And here's just some preliminary data again from this Eastern Pacific Ghostship transect, whereby we can see that the chlorophyll to carbon ratio corresponds quite well with the presence of these genomic biomarkers for nutrient stress. I think this is a very promising and exciting avenue whereby we can link omics with optical measurements done both in situ up to remote sensing of ocean column, and thereby extrapolate the variation in nutrient stress that we observe in situ to both the global scale as well as through time. And this example here is from a collaboration with Toby Westbury and Mark Barenfeld. Next slide. The last thing that where we think that all the data collected from Biogoship could really advance some of the scientific objectives from the Ghostship is that could we use microbial variation in microbial communities, especially in the deep ocean, as traces for circulation and mixes of water masses. And so for these different examples, I hope to have given some different ideas of how both we can use the core Ghostship measurements to get a better understanding of the biology, but hopefully also that a better understanding of the biology in the ocean, this can really advance some of the linkages between the elemental cycles, remote sensing, and also as proxies and novel traces for the circulation and mixing of water masses. Next slide. So we hope that Biogoship, by integrating biological measurements on sectional surveys, that we can really advance our understanding of plankton biodiversity and the biogeochemical role. And we think that this is there's incredible synergy by this link, because we can cover the global ocean within a decade with established techniques that will provide a high resolution measurement for both omics, optics, carbon stocks in various biological components, and also the elemental variation and the ratios, so that we can provide new ways of connecting the different elemental cycles in the ocean. This will leverage sort of the large existing investments in sectional surveys and actually sort of a relatively inexpensive way to get and rapid way to get a global view on biodiversity at high spatial resolution. We've started to collaborate with international partners, which we hope will greatly expand the sampling effort and spatial coverage. We plan to use established techniques so that it will be easy for other community partners to get involved. And we envision this strong integration between core Ghostship operations and the scientific objectives with our own. And I think one of the essentially most exciting aspects is that as we get started then and with a continued effort, then we can really get into a situation where we can observe biological changes, especially the anthropogenic forces of the biological communities on a global scale. So this was my presentation for today. And what I was hoping that we could spend the last time on is a discussion of the questions and what that you have, but also maybe you can think a little bit about two questions. First of all, how could you use these global data sets? And also, you saw sort of a list of some of the measurements that we're thinking about. But do you have ideas for other measurements that potentially could be integrated into this effort? And we would then be very happy to start a conversation about that. With that, I would like to invite Nicole and Harriet Alexander up on the stage.
Hi, everyone.
And then take any questions.
So I'm reviewing the Q&A right now, and we have a question. Looks like a series of questions, so we'll start with the first one. How close are we to understanding uncertainties in these new Biogoship measurements? And to follow on that, what can we do to make sure these measurements are intercomparable to a similar level as the core measurements?
Harriet, do you want to give that a shot?
Sure thing. So I can speak mostly to the omics side. And I think that increasingly we're seeing metagenomics being used at a global scale. And one effort that I think is very much worth mentioning, and I believe Adam mentioned it in his talk, is the Biogeoscapes effort, which is also coordinating an intercalibration effort. I believe that the proposal is currently pending somewhere. And I think that having these efforts happening in tandem will really help us gain better insight into questions of intercomparability across these data sets. Adam, do you have any other thoughts?
No, I totally agree that I think it's important to intercalibrate, but I think one thing that's unique, especially on the omics, is that we don't know the true answer. And so that makes it a little bit different from maybe a chemical, a single element measurement. And so it's more like you have to make sure that the data is comparable rather than it's not so easy to say which one is the right answer, which may also argue for a diversity of efforts in that sense. I don't know about Nicole, if you want to add on some of the other measurements, how far people are on that time.
Well, it's so much for, in terms of flow cytometry, there's been a lot done over the years in terms of some intercalibration between instruments. And we also have a few working groups that are working on at least establishing protocols for establishing metadata associated with these different samples and different techniques and how to present that. And that's actually an OCB working group that should be releasing a final report very soon on that from that community. It looks like we have a follow-up question here, or actually I'll start with, how would ship logistics work given the constraints of Ghostship on water budgets, berths, time at station, etc.? So I can start with the initial responses that we've worked with the current Ghostship, core Ghostship program to establish at least 1 or 2 berths on board. And so although that is a limited number of folks, we are trying to, with some of the equipment that you have seen that Adam presented, is a lot of it will be in hopefully an automated fashion, or at least in a flow-through system, such as the bio-optical packages. So a lot of these things will be set up at the beginning of a cruise and be maintained, but a lot of filtering will be taking place with regard to the omics side of things. And that will be pretty much the primary role of a lot of the folks that are the 2 or 1 member that will be on board. And Adam, if you want to add to that, or Harriet.
I can add that it is a very good point that there are definitely some constraints, especially with deep ocean samples, where that becomes a real problem, getting access to enough water because you have low concentration of particles and so forth. And I guess all I can say is that it's a bit frustrating to be out in an ocean full of water and not having enough water for the science. But I think that's sort of a general problem, not just a Biogoship problem. But hopefully we can work with the community to really get enough water for the science.
All right. We have another question here. In the way to measure and understand carbon, anthropogenic, Biogoship have or will have instruments to measure the O2 fluxes between ocean and atmosphere. Sorry, worded that correctly.
I guess I can answer that a little bit. That's actually a major objective in the core Ghostship project, is to understand the uptake of anthropogenic CO2. And so there's a lot of measurements of DIC and PCO2 and pH, alkalinity, etc., to understand the inorganic carbon system. And so I would say that that's already covered. That's not so much going to be our addition from the Biogoship, but that's definitely something that's already a major emphasis in the Ghostship operation.
Do we have any other questions from the audience at this time? Let's see, we have a few minutes left here as we conclude.
I think that's a question from Charlie.
Yeah, there we go.
Yeah.
Sorry, they're moving around a lot. So from Charlie, it's like, could you discuss the potential utility of Biogoship to plankton, food webs, energy flows, and from plankton to fish?
I can take a stab starting at that. I think there's a lot of utility. We are trying to, one of the figures that Adam showed earlier kind of showed the diversity of different approaches that we're using to measure different parts of the ocean food web system. And in particular, coupling omics with imaging and flow cytometry, I think will get us a pretty far way. And in terms of the connection to fish, we have begun working with Luke Thompson, who's trying to integrate a lot of eDNA type measures. And there was an initial discussion about the potential utility of integrating some sort of acoustic measurements, although that is still being assessed or investigated. So, Nicole, Adam.
So another thing I would like to add is that these are not process studies. So this will give us the biogeography of a lot of different stocks in the ocean. And so what I think will be interesting is that there will be a strong connection between what we do and what the modelers are doing, where we're going to work with modelers to compare what are the patterns that we observe versus what are the predicted patterns. For example, the size structure of the community could be an example related to what Charlie Stock was asking here. And so I think one of the big questions, right, is what is the effect of temperature versus nutrient stress on a lot of these biological processes? And so what we can provide from Biogoship is the statistical relationships, right? What is the size structure along gradients of temperature and nutrients? And by having a really high, a lot of samples from a lot of different unique environments, right, we can potentially disentangle these two variables, which are often strongly covariant, but not always, and thereby provide some statistical based suggestions of the linkages between, for example, size structure, carbon stocks in different biological compartments, etc., with these environmental drivers. And then through that, we can then compare to see whether or not various models will get the same versus different predictions. And we've already done that a little bit on, for example, using flow cytometry to give stocks of the picoplankton. And there you can see quite distinct patterns in the observed versus a model predicted distribution, for example, along gradients of temperature and nutrients. And so I think there's a lot of promise in the interaction between this type of data and the modeling community.
We have another question here. A major challenge for Ghostship and for this effort is to support analysis and synthesis of data. Are there plans for fellowship opportunities? So I think from the Biogoship effort, this is sort of just a starting point. And what I've always thought from this is that we would be in more collaborative and follow-up proposals that would bring in the community to help analyze some of this data at this point. And I would say that, yes, we will be only able to do so much while on board, but yet at the same time, integrating this and analyzing the data, I foresee there being many more collaborative and outputs in terms of other proposals that would then lead to analyzing this data. And with that, I'm trying to see if
I would also like to mention that we have been in conversations with OCB to host some workshops, and this is up and running, whereby we would invite people can apply to join these workshops, where we would have the data ready and have discussions about what are some of the new scientific questions that you could use this data to address and so forth, and thereby really facilitate a lot of community effort. However, we recognize the historical sentiment, and I think that's a general problem in the Ghostship community, is that you collect a lot of data and then you have to find the support to analyze it. And we will be in the same boat, so to speak, with the same problem.
We just have two more questions here. I don't know if we have time to answer them. And I think Adam touched upon it briefly. Beyond next week's workshop, are there any upcoming community events for Biogoship? So I know we have plans for OSM next year, I believe, if that's correct, Adam. And we are also considering, trying to get information out about the work, maybe through a publication that will be coming out shortly, hopefully. And we have a final question here. You showed Biogoship data indicating large differences in oxygen requirements for carbon cycling respiration. Does Ghostship have plans to quantify bioavailable carbon sources across regions? Or does Ghostship have plans? Adam, would you like to take that one as a concluding question?
Sure. So there's a lot of, in the current Ghostship, there's a lot of measurements of course, all the inorganic carbon stocks, as well as the dissolved organic carbon stocks. That's all in part of the core Ghostship objectives. What we would add then is a lot of details on the particulate organic carbon, and especially how the particulate organic carbon partitions between different biological components. And so I don't know if you would think of that as bioavailable carbon or not, but certainly what is the partitioning of the different stocks among the various biological aspects of the community? That would be something that we would add. But definitely we would also invite, I'd be happy to have people involved that look more at the more detailed chemical composition of the organic carbon, if that's what you're sort of thinking about. But that's not part of the current objectives.
With that, I think I'd like to thank everyone for attending and hearing about the Biogoship program. And hopefully with our initial funding, we will be putting out some data in the next year or so. And with that, I'd also like to pass this over and thank Adam and Harriet for attending as well, and our Ghostship team. I'm going to pass it back to Heather Benway, who I believe has a small little surprise for everyone.
Thank you. Thanks for a great conversation and some fantastic questions, everyone. I would like to do a surprise drawing for an OCB swag package to be delivered to the winner. I'm going to draw 4 names now, and then at some point later today, I'll draw another 4 names, but I'm not going to tell you when. So I'm going to start by sharing my screen here. You can all see that. All right. I'm gonna spin the wheel. Are you ready? Okay, the first winner is Patrick Gray. So as I call you, so Patrick, what you have won today, I'm writing your name down so I don't forget, is a fabulous utensil kit, which will be sent to you if you send me your address. So I ask that you all email me your address. May can drop my email address into the chat, please. And if you could send me your shipping address, I will send you a packet of materials that you can use to enjoy OCB. All right, I'm going to spin it again. Are you ready? Oh, Jamie Poulter. Our next winner, Jamie Poulter. Okay, here we go again. Meredith Meyer, are you still here? Hopefully you're all still here. Okay, and the last spin of the day, or of the morning, should I say. Rob Spencer, going out to you. And that is what I have for right now. Please send me your addresses. May has put my email address into the chat. And I will be happy to ship your goodies out to you. with that, I would like to get our next person up on stage for the next presentation, which I believe is Ann Kellerman and her Arctic Colors synthesis team. Hi, Ann.
Hi. Should I just go ahead and share my screen?
Absolutely.
Okay. I guess I can just start. So this presentation is about a new OCB activity called Coastal Arctic Biogeochemical Data Synthesis, and it is in preparation for Arctic Colors. The main folks working on this, organizing it right now, are Antonio Mannino, Peter Hernes, Patricia Matrai, Rob Spencer, and Maria Tzortziou. So first I'm just going to give an overview of Arctic Colors, what it is, why it matters, what's planned. Then I'm going to give a description of the OCB data synthesis activity. And then there should be ample time for feedback, comments and questions. And if there are anybody here that's interested in participating, particularly in the upcoming workshop in October. So first, Arctic Color summary. Arctic Colors is Arctic coastal land ocean interactions, a proposed NASA field campaign program to examine the effects of land, ice, and future change on the nearshore Arctic biogeochemistry. But first, some background. I'm just going to talk a little bit about the changing Arctic, and then rivers as indicators and transporters. But if you'd like more information about Arctic Colors generally, there's a website already up, and that's at arctic-colors.gsfc.nasa.gov. And you can go there for any additional information. I'm sure most of you have seen an image like this before. So the Arctic is changing, and there's disproportionate warming in the Arctic. 2020 was again the hottest year on record, and this has many implications, not limited to, but definitely including decreased sea ice extent and duration of color, decreased landfast ice, increased coastal erosion, and increased permafrost thaw. All of these things have impacts on the lives and livelihoods of human populations and all the folks that live in these areas. Also has impacts on the alteration of land-ocean transport and processing of organic matter and nutrients. The location of Arctic Colors is essentially bookended by the Mackenzie and Yukon rivers, and the nearshore environment and several smaller rivers in between. So the Mackenzie and Yukon River are the 4th and 5th largest Arctic rivers by annual discharge, the 18th and 22nd globally, with mean annual discharge 16 and 208 cubic kilometers per year respectively. And both Mackenzie and the Yukon watersheds are both largely underlain by permafrost. So just to give some examples of why it's so important to get started, there have been many notable events in and around Alaska in between 2014 and 2019. So up in Utqiagvik, they've already had like 5 warmest winters in 2014 to 2019. There's been flooding both coastal and inland. Record heat in Anchorage in 2019. So there's a lot of reasons why it's so important to work. Next, why rivers? Rivers are modulators of land-to-ocean transport of organic matter and nutrients. Rivers integrate inputs and signals from the catchment, and changes in export and processing will have implications in the nearshore coastal zone. And then freshet is also changing. So changing patterns in precipitation and temperature are altering the dynamism of freshet. This will likely have implications for the amount and timing of land-to-ocean carbon transport. So we need to set a baseline of current conditions to understand the extent of future change that we want to look at with Arctic colors. So Arctic colors, Arctic coastal land-ocean interactions, is a proposed NASA field campaign program that aims to quantify the coupled biogeochemical ecological response of the Arctic nearshore system to rapidly changing terrestrial fluxes and ice conditions in the context of environmental and climate change. And those can be looked at as short-term and long-term change. The overarching science aims are to establish the effects of land, ice, change on the nearshore Arctic biogeochemistry. And this is a more detailed study map. You can see the Mackenzie and Yukon River bookends here. And then there's a lot of smaller rivers in between, Colville, Kobuk, etc. And then also the nearshore coastal environment, which is this pink shading here along the coast. The Arctic Color Science Plan. We have the summary and we're making minor adjustments to the plan, but the plan is to integrate coupled land-ocean ice models with remote sensing data and field campaign observations to forecast and hindcast carbon fluxes and productivity across the land-sea ice continuum. Next, implementing emerging satellite and airborne remote sensing data fusion capabilities to integrate across multiple spatial and temporal scales. And then the third item is utilizing field campaign observations to evaluate and improve NASA satellite ocean color retrievals in the coastal Arctic. So that's Arctic colors. For now, this new OCB activity is the Coastal Arctic Biogeochemical Data Synthesis. And this is really to kind of set the state of the art with what data we have and what exists right now. So the OCB data synthesis aims are to provide a comprehensive synthesis of existing ocean biogeochemical data in northern high latitudes. Primarily, this is going to include data on carbon stocks and fluxes, along with optical proxies, for example, absorption and scattering, and then any and all appropriate metadata. Other biogeochemical variables, for example, nutrients and processes like will be included where possible. Another aim is, the main aim is to collect and combine existing in situ data and modeling results in satellite data sets. And part of this will include resolving differences in reported data due to methodology. And then from that, we're going to create and host a new synthesized database with all of this data that we've collected combined. And from that, identify data and knowledge gaps from the data that already exists. So the timeline for all of these activities is quite lengthy. There are a few projects already in place, like the above project Arctic Boreal Vulnerability Experiment and the Beaufort Lagoon LTER. Those have both begun. There's a number of pre-Arctic colors projects down here that started in 2018 and are ongoing. The OCB data synthesis activity is starting now, 2021, and will go into 2022. And that's so that's all in preparation before the next solicitation phase. So what are we looking for? We're looking to recruit interested participants for the interactive workshop to be held in 2021. Ideally, participants should contribute data, for example, on carbon stocks, fluxes, or optical proxies. And then we know that there are several existing databases and data sets and repositories, and part of this exercise is to synthesize these data. So we welcome any databases or data sets to look at. And yeah, so those are the main things we're looking for. And with that, I'd like to acknowledge many people, including Heather Benway and the Ocean Carbon and Biogeochemistry Program, Paula Bontempi, Laura Lorenzoni, and the NASA Ocean Biology and Biogeochemistry Program. The NASA Review Committee that reviewed and provided really useful feedback on the science plan. Participants in the 2016 Arctic Colors Workshop. And then all the additional Arctic Colors Scoping Study PIs and the many collaborators and co-authors. So with that, if people have questions for the panel, if we can get the rest of the panel up on stage, that would be great. And if anybody's interested or has questions, contact information is listed, and then the website again. Okay. Should I close the slides?
I think we can keep the slides on just to, yeah, in case people have any questions.
Could we get Antonio Mannino on stage as well, please? If he's here. Yep, he's here.
Antonio, you should be able to activate your camera and microphone at this time. If not, we'll try resending the invitation to you. Stand by. We're going to try to bring Antonio back up here. Hold on one second. There we go.
Great. I see that there's one question already. Will there be a specific call through either NSF or NASA for science to support Arctic colors? And yeah, there will be a NASA solicitation. The first NASA solicitation will be for the science definition team to work on the science plan for Arctic colors and the implementation plan for Arctic colors. And I believe this should be coming out sometime within the next year or so. Are there specific instructions on how to contribute data sets? Antonio, would you like to take that?
Certainly. Good afternoon or good morning. So Ann Kellerman is leading the charge on this aspect. I would suggest sending her an email regarding your data sets. You know, what the measurements include and just general geographic location. You can also CC myself and any of the other folks here on stage as well. But Ann and CCing the rest of us would be a good starting point to reach out. Ann, anything you want to add? Go ahead, Rob.
I was just going to add to that, Antonio. So we have a set kind of format for that, Miguel. So if you want to reach out to myself, Antonio, or Ann, we can send you the template if that's easier, or if it's just easier for you to direct us to where your data is in a repository, we also kindly have a couple of interns from NASA that can then go and work through that. So just let us know whichever is easiest for you. But I think, yeah, as Antonio said, the best place to start is emailing Anne, and we can point folks in the right direction if that helps. And her email's on screen. It's on the bottom right-hand corner in mine. It's quite small. Thank you, Heather. And Heather's just posted it in the chat as well.
Great, thank you, Heather. And Miguel, as Anne mentioned, this is just the first steps towards synthesis. And for this specific first effort, we will be focusing on the synthesis of carbon stocks and fluxes along with their optical proxies. So we're looking at absorption, scattering properties, and metadata. But other biogeochemical variables, nutrients, primary productivity would also be, of course, very relevant. And it would be great to see what data products, what parameters the community would be able to contribute. So just let us know what data products you might have, and we will get back to you. There are more questions on the Q&A. Patrick is asking, what kind of timeline do you expect? When might fieldwork start, and how many years, any data collection throughout the Arctic winter?
I'll take a stab at starting. So as Maria mentioned, the first step is an implementation, is for NASA to solicit implementation team, would be just a group of scientists and stakeholders in this study region to develop a plan for the field work and other and related aspects to the effort. Identify the gaps in modeling and remote sensing needs. And our expectation is that NASA would solicit for fieldwork to begin in the probably 25 timeframe, realistically. So solicitation would be somewhere in the 2023 to 2024 timeframe. Again, this is all TBD. NASA currently is supporting a large field campaign that you probably heard throughout the OCB program called Exports. And so NASA can only support one large-scale field campaign program at a time. So it would be we would be the next one in line. Oh, and so to address the other part about collection throughout the Arctic winter, that is what we recommended through the science plan. We collectively felt that was important. But again, the implementation team would provide further guidance on that aspect.
There was another question we missed at the beginning. Charles was asking, you mentioned a goal of ultimately modeling Arctic carbon and ecosystem dynamics across the land-ocean interface. Could you speak a bit more about the strategy for this? And yeah, Charles, we know that there are some ongoing efforts to model physical and biogeochemical processes in the coastal Arctic. And as part of this synthesis effort, we would like also to include the modeling component. In addition to field data sets, we would like to see what has been done so far in terms of coupled hydrodynamic biogeochemical models in the coastal Arctic. Where are some of the major gaps? And what data we would need to collect in order to start addressing some of these gaps in models. And in addition to models and field data, of course, the remote sensing algorithm development as well. So any groups who have been working towards the development of coupled physical biogeochemical models for the coastal Arctic land, ocean, ice models, we definitely like to include these efforts here in the synthesis effort.
there's a comment from Erica in the general chat. She notes that I think DOE sponsored coupled models in the Arctic recently, maybe in the last 5 years. Yeah, and I think that's absolutely true from what I understand and is ongoing. And I would hope that some of the data that could be generated from this would be of use to those as well. And so I'd see an active area for collaboration there.
Yeah, Jamie Poulter had another question. What role is envisioned for autonomous data collection in the Arctic?
I think we believe that that's an important contribution to the overall effort. Obviously, there are limitations on what can be accomplished from ships or boats or from just specific sampling events, continuous records of various optical and proxy measurements, as well as physical parameters, are quite important throughout the year. There are a number of groups who have developed approaches for collecting continuous autonomous data. Victoria Hill, among many others, including Sam Laney, currently and others. So we expect those plus other platforms, whether it's gliders or other types, would prove quite relevant and critical to the success of Arctic Colors.
There's another question in there from Miguel as well, in terms of how is the coastal domain defined, water depth, distance from land? And I might direct that one to Maria, because I think she spent a bit of time thinking about that one in the proposal.
Yeah, I mean, this is something that we provide some information in the science plan on Arctic colors, but this is something that we could revisit as part of the science definition efforts and also the implementation plan. And Arctic colors is indeed very much focusing on the land-ocean interface and the exchanges between the land and the ocean, but we absolutely want to quantify, parameterize processes in the coastal ocean as well, in the ocean domain. And it's something that we were thinking that we could revisit with a science definition team in terms of the freshwater end member and the marine end member.
I would add that Anne showed a notional study area in one of the maps that she had on her slide set. It's also in the science plan that's online. That the link was provided by Heather and the website by Maria. So that provides just at least a notional context of what we thought would be manageable. But again, the implementation team will further refine the study area.
Something that will also depend on the synthesis effort as well. We want to see where we have existing data sets, where some of the major gaps are, and this will be part of the decision for the science implementation team to make the final decision on where Arctic colors would be focusing on in terms of the field observations and the in situ data collection.
And that's something that's come up in our group's sort of discussions. And so I'd encourage everyone listening to this to tell us if we're wrong. This is the chance for this, right, through the synthesis effort, is that we're aware of a lot of data on the riverine end, on the freshwater end, and we're aware of a reasonable amount of data on the blue water oceanography end, if you like. And there's kind of this gap in between. So if anybody's sat on a large data set there that we're not aware of, especially in this proposed study region, please let Anne know in the coming months so we can include it, and that can then help focus this effort going forward, if that seems sensible to everyone.
For example, you will see in the science plan, we define the areas from the Yukon River to the Mackenzie River, that whole coastal ocean domain. And in terms of freshwater and marine, and members, we mentioned the head of the tidal influence all the way to the coastal shelf. But this is obviously a very large domain, and narrowing it down would require this information from this synthesis effort and other results from ongoing studies, and the discussion that will happen within the science definition team. I think we have another one under Q&A. Will you envision ocean acidification in the data synthesis efforts?
So, yes. So, PCO2, DIC, and related ocean acidification variables, parameters, total alkalinity is part of our list of measurements of interest.
Do we have any other questions? I don't see anything else under Q&A. Thank you, Patrick. Yes, we're very excited about Arctic Colors, and we're very excited this is happening after many years. People who have been participating in OCB meetings know that we had the kickoff Arctic Colors meeting back in 2014. So this has been an ongoing effort by many members in the community who have been contributing in the discussion. They've been contributing with data sets. They've been contributing with information for the science plan. And we're very excited to see that we're moving forward with a synthesis data effort now, and with the science definition team and implementation team within the next year or so. So
I think that's a great point, Maria. I think that it's important for people to realize that field campaigns, these large initiatives like this, like exports, these take about a decade to put together, and they take community champions. This really is grassroots stuff, and a lot of it starts on a napkin at a meeting over lunch, or and it builds, and there's a lot of iteration and community feedback, and that's how this works now. And if you have a great idea, start talking to colleagues, start getting a session at Ocean Sciences or get a session at an OCB meeting. And let's start the discussion, because it really does. It takes a long time, and it takes a lot of work and a lot of effort. But in the end, you know, it's hugely transformative in terms of the multi-platform data sets that come together and the insights that come out, the publications, the mentoring and the early career development that comes as part of it. This becomes somebody's graduate student experience or postdoc experience. And I just think that this is the way science should be done. And it really is fantastic to be watching it be done in real time. OCB's been around for a while now, and we've gotten to see some really cool stuff come out. So. All right. Well, I want to thank Anne for a great talk, and thank our panelists for being here and answering all of these questions. There certainly is a lot of interest in this, and we'll continue to keep you posted. This group will have a web page, and we'll be getting their work underway, so we'll keep you posted. And please be reading the Arctic Color Science Plan, keeping tabs on the website, and looking out for the science definition team call that will be coming out. With that, maybe I should give you, I guess no, it's 1:29. So I guess we can bring our next speaker up on stage and the panelists. I want to thank you all again. Our next speaker will be Anne McNichol, who will be talking about another OCB activity highlight. Welcome, Anne.
Hi.
She's probably a couple miles away from me. Same with Roberta, both also at Woods Hole Oceanographic.
One more.
Is Bob joining us today?
He is. He was in the practice
Super.
session with me. So
Great.
To hear it. No.
Bob is on
There's
stage,
Chase.
and you can turn on your camera and microphone, Bob, when you're ready.
Oh, okay.
Here we go.
Oops. They'll get there eventually.
Bob, you may need to refresh your page in order to come back on and turn on your camera and microphone.
At least he's not falling off the stage. I went to a talk where a speaker literally fell off the stage, but there was a space between the platform and the screen, and
oh my God. It's much better to fall off the stage virtually, I think, to fall off physically. Here's Bob.
Yeah.
Hello.
Yes.
Welcome.
I refreshed and it took me all the way out of the program.
Oh.
I made it back now. Let's see if I can get to where I can hear myself. I can hear all of you.
We can hear you.
We hear you great.
Okay.
And Anne,
Yeah.
will you be sharing your own slides?
Yes, so I'll
All
just
right.
hop in.
Great. Yep.
Okay. Let's see if I do this correctly. All right. Oh, actually, question. Do we know if there's anyone else in the room?
7
Everyone's in the
people
room
here.
and they can see your slides. You
Okay,
can
but there
begin.
are people here. Excellent. What did you say, Chase?
You may begin. There's 47 in the audience.
Okay, excellent. Thank you. I'm here to talk about a program that OCB has just funded recently, where we're hoping to discuss how to ensure that the results for measurements of delta C13 and C14 in dissolved inorganic carbon remain high quality and get even better in the future. Our hope is to convene this in the fall of 2021, this year, either in person, virtually, or hybrid. That will be determined in the future. My colleagues are Roberta Hansman, Bob Key, and Rolf Sonnerup, who is unable to be here as he is collecting salmon in Alaska right now. We've been studying, the four of us are studying dissolved inorganic carbon in isotopes as part of the global programs, WHOI, CLIVAR, and Ghostship, and beyond. the isotopes are powerful tracers of the carbon cycle in ocean circulation. To date, there are over 30,000 measurements available in the databases, but to date, there is no recognized reference material, and we're hoping to change that, and starting with the workshop that we will convene. Now, the measurements are generally comparable, but they would be much better with a certified reference material. For the global programs, there have been mainly 3 labs doing the isotopic measurements. NOSAMS, which is the National Ocean Sciences Accelerator Mass Spectrometry Facility housed at Woods Hole Oceanographic. University of Washington through Paul Quay's laboratory, and one of the Japanese laboratories, I believe it's JAMSTEC. I'm sorry, I didn't check that out before I got on stage. And the laboratories doing that are currently ensuring the quality of the isotopic measurements through IAEA and NIST isotope standards for the accelerator mass spectrometry, stable isotope ratio mass spec measurements, and the newer instruments that are used to measure carbon-13, such as the cavity ring down optical systems. Also, laboratory produced secondary standards ensure reproducibility over specified periods of time, such as if you collect 100 liters of water and make X number of bottles of a standard and use that over time, you get to know the reproducibility of your measurements. Deep water comparisons and interlaboratory comparisons. With respect to the deep water comparisons, the hypothesis is that there is, over a couple of decades, for many parts of the ocean, there is no significant difference over time in the measured value. And this is actually how people are able to use the results from the 3 major laboratories for C13 modeling. You can do multiple linear regression analyses to come up with an offset or a correction factor to apply to the labs so that they all have comparable values. However, it would be much better if they were all comparable before you put them in the database. So just to, early on, 1994, at the beginning of the WHOS program, NOSAMS was having samples collected in the Pacific Ocean, and we used results from geosets. These are depth profiles of dissolved inorganic radiocarbon in the Pacific Ocean seawater. And our hope was that below 750 meters, we would get results, basically identical results. And these data show that, yes, in fact, we did, and even shallower at some instances. So the Austin's results, I think I said, are from the GeoSix program in the 1970s. And our results are from the 1990s. For C13, similar, comparing samples taken a year apart in the Indian Ocean. These samples were close to each other, not spot on, but you can see that the samples collected in 1994 for NOSAMS and those collected for UW for C13 now we're looking at dissolved inorganic carbon 13, have very good agreement at sites close to each other. So that's how it was done in the early 90s. By 2001, 2 decades ago, there were enough laboratories conducting these analyses that an intercalibration was started. What we have here is on each graph, the left-hand axis is an identifier for different laboratories. And you'll notice that this one goes from B to G, and this goes from A to F. So 4 samples of gas, of homogenized CO2 gas in flame-sealed tubes were distributed to each of the participating laboratories, and 4 bottles of homogenized seawater were distributed to each of the laboratories. And Delta C13 results were reported back and are shown here. This work was never published. I no longer recall who the other laboratories were. The point of the graph is that there are offsets. Note that the x-axis only encompasses about a little over 0.3 per mil in delta C13 space. But for oceanographically useful results, you would like the results to agree within plus or minus 0.05 per mil, which is not being hit here. So it's known that there are offsets. People have been working with the offsets. It's not surprising that there are offsets, but if we could get away from that, that would be great. More recently, Chang et al. Published a paper in 2019 where 16 laboratories measuring DIC13 participated, and they were given bottles of seawater, which are in the top graph, the blue squares, and bottles of a reference material, which is Andrew Dixon's DIC, certified reference material. It is not certified for isotopes, but their hope was to use it for that. And you can see that there's a fair amount of scatter in both the blue and the black dots, but when they take the data from the blue squares and assign a specific value to the Dixon seawater and then correct everybody's results, there's a much greater agreement amongst all the results. And in the average, going backwards. It's interesting. The average difference, standard deviation of these results is plus or minus 0.06 per mil, which is very close to the 0.05 per mil that the global ocean observing systems defined as the desirable spread for C13 results. So this shows that a reference material can really help, and that's excellent. Another point I wanted to make was that C13 is getting a reference material for C13 is almost more important than getting one for C14, because C14 results are corrected for the spread in the C13 isotopes already. And there, aside from major methodological issues, there shouldn't be quite the discrepancy that you'll see in the C13 results. But that's my opinion. I don't have data to back that up. So that's the rationale for the workshop. The topics we intend to cover are, just broadly speaking, the need for standard isotopic methods. It's good to have everybody making these measurements using all different methods to come together and say, if you use this method, this is the best way to do it. And from ship to shore to database. So we'll look at the results from prior interlaboratory comparisons and hopefully find some more in people's lab notebooks tucked away somewhere. Discuss collection preservation methods, the different methods that are being used for analysis. There are shipboard methods now for Delta C13. Reference materials, which I'll talk about a little bit more, and data issues, data management and procedures for reporting uncertainties. So I wanted to point out that we have an opportunity to piggyback on the work that CO2 and seawater reference materials programs are talking about. They're looking for the future. Andrew Dixon has been doing a spectacular job providing the DIC community with reference materials. And from the C13 comparison study that I just cited, it would be wonderful if we could certify some of the batches that he makes for carbon isotopes. And we're starting this discussion, and we'll continue it through the workshop. And the Dixon program is not going away immediately. It currently has 3 more years of funding, so this is a good time to start working all of that out. As I said, we're hoping to have this workshop in the fall, and there is a tiny URL that you can fill out to express interest. It's the link is available in the chat. It's just asking you for your name and your email, basically, so we can find out how many people are actually interested in this. And the workshop will have products, so if you attend, you will be given homework. And that's important to know, I think. So that's all I have, and I'm happy to things up to discussion right now. Questions? Comments? Oh, well.
Oh. And under Q&A, we've got a question.
Right.
Yeah, I can read that out. It's from Andrew. What is the desired standard overall uncertainty in delta C13 for any putative seawater-based reference material? And note that this involves uncertainty contributions from 3 areas. The measurement, and I cannot read the rest.
Yeah, I don't have that either. So, you know, this is, I don't, measurement technique. Well, I do know that Paul is able to measure things to plus or minus 0.03 per mil. And so that would be the bare minimum for what we would be looking for. That's my answer. Anybody have a different answer?
Well, I think that would be an awfully good place to start if we can get there, and then it'll be like the DIC measurements. The C13 measurements should improve with time, and as they do, one would hope the certification precision could get better as well. Yeah, 0.03 to, gosh, what are we at now? 0.3 more like.
Yeah. So wait a second. All right.
There's another comment, in the questions that NIST can probably help with these isotopic reference materials. So that's also another opportunity.
That's great to hear. I keep forgetting to point out that the one issue with radiocarbon is you have to make a radiocarbon clean standard. It shouldn't be difficult, but if you're not paying attention, it may not be great. So
there's another question in there if we can use C13 isotope methods to determine for POC or DOC in seawater as well, and what method we would recommend if that could be used. I think this is a very different reference material, so we'd have to look for something different for organic materials. That's my take on it. But
yeah, I'm not sure what the question means. Are you asking if you can measure C13 in POC and DOC? If so, the answer is yes, and people do it. The precision is, of course, much worse than for DIC. DOC is a very challenging method. But Roberta would know more about that than me. So.
Well, and of course, Ellen has published on her techniques. Does anyone make any more measurements than she does on the?
Our line has been very, very busy here at
been
NOSAM.
busy as well.
So we've been running a lot of samples for clients for VO14. Yeah.
But I think we can safely assume that the reference material we're talking about for DIC in water clearly would not be the same as one would use for DOC or POC.
No.
It couldn't be.
Actually, I wanted yesterday or 2 days ago when Andrew Dixon was talking, he was talking about how grease is fairly inert, and I did want to point out to him that we once had a bottle of seawater with a blob of APAs on grease floating in it, and it affected the delta C13 significantly because of storage over time, and it was about 2 per mil off what one expected. So something oxidized it.
Yeah, and to comment on DOC versus DIC, this reference material and other DIC seawater samples are preserved with mercuric chloride typically, and that is a no-go for our DOC reactor. It can poison react with the UV, and it's not an ideal preservation method to make a DOC measurement. So those are pretty conflicting.
Yes. That's because the DOC oxidation is a UV oxidation, but just the fact that you use grease for stuff is also a big no-no. So Andrew has said that he knows that the Keeling group has a measurement precision of 0.01, but the uncertainty is likely larger. Yes, so when I'm talking about 0.03 per mil from Quay, that's from what I like to say from ship to shore. That's everything which, as you know, you always want the instrument precision than much tighter than the actual method precision.
And regarding that first question that's just been posted from Michael, what is available from NIST?
I don't know. Maybe Michael can tell us. I don't know what oceanographic CRMs are being worked on. I'll give him time to answer if he's still there. So there was I'm not sure I understand the question about measurement of POC and DOC. The methods for measuring those are quite different from the methods for measuring dissolved inorganic carbon. There's a lot of literature on the techniques used to measure those, and I don't know, maybe Roberta can point you to some specific references, but
yeah, I can put my email in the chat if anyone wants to shoot me an email, or if Delito wants to shoot me an email, I can answer any questions.
I did not introduce us. So Roberta is the staff chemist at the National Ocean Sciences Accelerator Mass Spec facility. I don't know if that's the title. That's the title I used to have when I worked there. And I'm still at WHOI, sort of closing down my career. But I've worked with carbon isotopes in the ocean for a long time. And Bob Key is in charge of all the data in all the global databases. So he's a oceanographic chemist at Princeton University, emeritus, and has been responsible for helping, for putting the GLODAP database together, and helping make sure that most of what's in the database is as tight as it can be. You're working, so Michael Winches, we are working on metacresol purple for seawater pH. We have a long experience with isotopes and all kinds of CRMs generally. We're looking to expand our offerings in oceanography. That's the short answer. Well, I hope you're filling the tiny URL out so that we can invite you to the workshop when we have it. So, it sounds like your participation would be an excellent addition. So.
Great.
Okay. And if you know of other people who would be, for whom it would be a good idea to attend this workshop, please either email me or Roberta or Bob. I'll put all of our emails in the chat. Please pass this information on. We want to have the best workshop possible. Oh, Heather has taken care of things for me.
And would you think the workshop would be, well, let's see, better stated, what do you think the odds are that the workshop will be in person? Clearly it's a guess.
I don't see how we'll be able to get everybody here. I mean, if there's anybody in Australia or New Zealand who wants to participate, there's no way.
No way certainly no way to make it global.
You know, I don't know how a hybrid workshop, if it's a possibility, and what it would mean. You know, that's definitely something over the summer we really need to look into and watch what's happening. I mean, they're talking about a fall surge, so that's not good news. So I know all of us would far prefer it to be in person. But I'm pretty sure everyone at this conference would prefer this conference to be in person. So. Heather chats that hybrid is something we're thinking about a lot. So we will be in contact with Heather and hopefully plan on she's correct in pointing out that not everyone is going to be comfortable going to an in-person workshop. We don't want to exclude those people. And I guess hybrid is going to be the wave of the future. So as our workshop says, improving results in the future, we'll have to improve the workshop results in the future as well. But we only have 2 minutes left, and I don't know that I thank you for sitting through all of this, those of you who are still here. I'm looking forward to the workshop. I think we have a great opportunity to really improve these measurements. Do you guys have any parting words?
Just say that there's a ton of evidence, not only the CRMs that Andrew made for the carbon community, but we've had similar experience with the nutrient CRMs that are reference materials that have been made in Japan recently. They all have significantly improved the quality of the data, and when you start putting together these big databases, that's when you really notice it. If you're only using data from one lab over a short period of time, then it doesn't make much difference. But almost everything that most of us do nowadays is global, and that means you have to put different people's data together, and as soon as you do, then all of these sorts of topics become very, very important. It's very nice not to have to make adjustments to the data, especially when you don't know which way to move them, which was the case with the silicate in the nutrients in the recent Japanese-US experience. Both groups were turning out just fantastically precise data, and we had no idea which one was right. There's no way to tell.
Yeah, and Bob, just to piggyback on that, Andrew asked how many reference materials we would need in a year, and I would guess we could look back at the databases to kind of see how many groups are contributing to these types of measurements to get an idea. But I think we talked about this, the three of us recently. We don't really have an idea what demand would be until we kind of get more feedback from the community.
Yes, and there are a number of laboratories that are just ramping programs up across the world, and that is hopefully at the workshop we'll be able to track those. And there's more and more C13 since the optical methods are going to broaden the base. So
yep,
stay tuned.
Yep. Very good time to be doing this.
And a very good group to be doing it. So thank you.
Thank you.
I want to thank our speaker, Anne, for sharing. This was a great idea she shared with me, over a year ago, and
yeah,
I'm so glad that a proposal came about and it was selected enthusiastically by the SSC for funding. So we're looking forward to it. We will certainly keep you posted as this effort develops, and we will also put that tiny URL link in the next e-newsletter, but it's also in the chat. So please do fill that out if you're interested. That really helps us kind of target communities that would contribute to this. So with that, I want to thank you. Thank you, Bob and Roberta, for fielding our Q&A and for being part of this great effort.
Mm-hmm.
And I guess we're moving towards wrap up and another surprise drawing too.
Woohoo!
Woohoo!
Thank you, Heather.
Thank you all so much. Now, as soon as I get on stage, everybody leaves. I'm trying not to take this personally. So I'm going to share my screen.
Hey, Heather, sorry to jump in on you. Somehow we lost your microphone there, like right at the moment you started to share your screen. Do you mind just turn it off and back on again?
Sure.
Oh, there we go. We hear you now.
Great, thank you. It's like the IT crowd. Have you tried turning it on, off and back on again? So could we bring Victoria up on stage, Mike, if she's still here? So Margie is the Scientific Steering Committee chair and Victoria is the vice chair. Victoria is the incoming chair. She will become the chair at the end of this year for the following 2 years. And so it's kind of fitting that we started the meeting and welcoming with Margie, and now Margie can't be with us today, and Victoria is taking the torch. So she is going to be here with us as we say our farewell. I really want to start with just a big thank you. I can't say thank you enough, and I think it's really important to say thank you because efforts like this don't come together easily. So I'm going to just thank a few people specifically. Certainly, I want to thank our Scientific Steering Committee and session chairs and speakers for the fantastic plenary sessions, which were a real highlight for this workshop. And I know it was a lot of extra work to put these talks together in advance and add closed captioning and all of the extra work that went into that. But I want to thank you for that. I want to thank you for the great questions that you came up with that we discussed during the breakouts. And those whiteboards, that all of that content has been saved so that we can use that as a springboard to move forward. I want to thank our poster and networking presenters. I know that, you know, it's not easy to take on a new platform. You know, our poster presenters not only prepared a poster and a recording in advance, but then they were here and present for an interactive session and made additional versions of their posters to share. I want to thank our program partners and agency managers who gave their time at the networking tables to share information and talk with people. I also wanted a quick reminder and shout out for all those posters. There is going to be a poster gallery, the virtual poster gallery, which will be open all through the summer. So you have plenty of time to go back in if you missed a poster session and watch those, look at those posters and watch the presentations. I want to thank our event hosts. ePoster Boards has just been amazing. They are, you know, the main person that we worked with is Mike Elliott, and he really helped put this whole meeting space together for us and really walked us through day by day. We had a lot of meetings. Just getting this all together. So I want to thank all of the people from ePoster Boards, Catherine and Eduardo and Chase and you know, everybody who was involved in helping pull this off. You were all just such a great team to work with, and I will be a champion for ePoster Boards from now on because it's been a great platform. I believe a year ago, ePoster Boards wasn't even doing this. It was more of a physical presence where they would provide meeting support, and they've transitioned this whole operation and just really turned it into a pretty amazing platform. My dream team, May Mahegan and Mary Zawoski, couldn't do this without them. This is how we get this done. We all bring different things, different knowledge and skill sets to the table, and it's been really great to work with them. And of course, our sponsors, NSF and NASA, this program wouldn't exist without you. So I want to thank our program managers from Chemical and Biological Oceanography and NASA Ocean Biology and Biogeochemistry, and Woods Hole Oceanographic, not only for hosting the project office, but providing an endless source of stimulating ideas and conversation. I am constantly talking with OCB scientists who are wandering the halls of WHOI. You know, OCB certainly casts a wide net. It's an international network, but there are a lot of them at WHOI, and I get to talk with them a lot, and it really, it makes my job a lot more fun. I want to encourage you to stay involved, especially if this was your first meeting. The first step in doing that is really being informed. Please be informed. Subscribe to our monthly e-newsletter. It's worth it. provides job opportunities, updates about our program and what activities are going on, what our partner programs are doing that might be of interest, education outreach, student opportunities. It's just a really great compilation of information that comes out once a month. Get on our website and look around. Small group activities, our annual summer workshops, scoping workshops, which are topical workshops, leadership, anything you can think of. It's on our website. So take the time to go to usocb.org and explore. Follow us on social media. We have a YouTube channel. We are on Twitter, and we share a lot of news via Twitter, actually, between e-newsletter issues. So I really encourage you to be informed. That is the first step in staying involved with the OCB network. I talked on the first day about this being a bottom-up grassroots program, and it really, really is. It's community-driven. It's really important to share your science. We have so many different ways that you can contribute to the OCB network and really get something from it. A lot of the early career folks who I started interacting with years and years ago have come up through the OCB network, and now they're giving plenary talks, and they're leading, and they have bringing their own students to our meetings. And this has been a really great experience. We have, for every e-newsletter issue, we have opportunities to put out a science highlight. So if you have a paper you just published and you want to get some more attention to your paper, you can do a science highlight, just share that with us. You can propose an OCB activity. We have an activity proposal solicitation that comes out usually late summer every year. Propose a summer workshop session. We put out in our newsletter a call for ideas for plenary sessions. Nominate yourself or a colleague for OCB leadership positions, including the scientific steering committee. Participate in OCB activities like you've already done. And to follow up on that, there are quite a few that are upcoming. The GoBGC workshop is coming up actually Monday, Tuesday, and Wednesday, and you can still register. If you go to our website, you can register for that workshop. It is all virtual. I strongly encourage it. There are a few other things coming up. We talked about the Biogeoscapes workshop. That was the very first plenary session that we had, actually mentioned this, and a lot of the conversations we had during that plenary session will help inform some of the topics that we'll be discussing. This is really a U.S. planning workshop for a large international biogeoscapes program. A lot of other nations have already had their planning workshops, so this will be the U.S. planning workshop, likely taking place in November 2021 here in Woods Hole. It's likely to be a hybrid model depending on people's comfort levels with travel. There is next month in July 12th, the Biogeochemistry of Mixotrophy. That will be the Mixotrophs and Mixotrophy Working Group. They have meetings bi-monthly, and you can join. They're open. Usually the first part of the meeting is open to everybody who wants to come. There's a presentation or two. So this will be the next one, and that will be the topic, July 12th, 11 a.m. You'll see an announcement in the OCB News about that and on Twitter. The daily to decadal ecological forecasting along North American coastlines. That's a scoping workshop that we are co-convening with the U.S. CLIVAR program. That will be taking place in April 2022, also in Woods Hole. We don't usually have all of our workshops in Woods Hole, but it just so happens that these next couple are here in Woods Hole. And finally, next year's OCB Summer Workshop, which we hope to hold in person, will take place June 20th to the 23rd, 2022. And we hope to see you there, and we hope to also provide some form of hybrid options in order to accommodate broader participation. We usually, at the very least, we webcast the meeting. Late breaking, I just added this as Anne was talking, carbon isotopes in the ocean, really planning a planning workshop for for the future, for a future intercomparison, to express interest in this workshop, there's the tiny URL. We'll be sure to, that's in the chat, and we'll be sure to keep that coming at you on Twitter or in our e-news or wherever we can. Finally, I really want to keep these conversations going that we started at this workshop. These are important conversations, and they don't have to end here. I think that people provided a lot of great feedback when we had our discussion about the diversity survey and how OCB can support broader participation in the ocean sciences and the OCB network. These are some great ideas that you provided to us, and we would really love to keep this alive. So I strongly encourage, this URL will be available. This Jamboard will stay open. There's also an early career Jamboard. The early career community is really strong, really flourishing in OCB. And I would really like this Jamboard to stay alive. I would love these conversations to continue. There's also a Slack workspace for the early career community that will also remain active and open. So I strongly encourage you to explore these resources. There are actually, on the workshop website, you will see an early career page and a JEDI page, and all of these resources can be linked right from there. So please keep those conversations going. So the workshop survey, we will be developing a workshop survey and distributing it within the next week or so. It will go out to the entire workshop email list, and I really strongly encourage you to take the survey. We cannot do this without your input. And this was a fairly unique year for our workshop. This is the first fully virtual OCB summer workshop that we've ever done. So please, please, please take, we will make sure that it can be taken in 5 minutes or less. Please take the time to give us your feedback. In the meantime, we have another short Menti poll for you. So I have put this here, and May is going to drop the link and the code into the chat. And I'm going to start sharing my Menti screen. So hold on one second.
Whoops, it looks like Heather got lost there, and I'm hoping May has put the link in the chat. Oh, good.
Okay. Can everybody see that? I hope we can. We'll look and see if we see some results start coming in here. Data synthesis, absolutely. Finding funding for data synthesis. We invest a lot in our observing systems and in our process studies, and it is really critical to make sure that we're getting our return on our investment in investing in data synthesis. And that's part of the Arctic Colors presentation that you just saw. More data synthesis. Holy cow, everybody wants data synthesis. We've had 3 responses. Do we only have 3 people left on here right now? Come on. JEDI. Data synthesis. Helping early career scientists survive COVID, absolutely. Well, this meeting was really structured around providing a platform for interaction. Can anybody provide some specific feedback on data synthesis? What kind of data? Any high priorities or exciting, time sensitive? Increasing diversity, improving collaboration. Yes, getting past COVID. It has definitely put a monkey wrench into our Jedi. Yes, absolutely. We have some great ideas on that Jedi Jamboard that we'd like to follow up on. Strengthening code and data sharing, absolutely. Collaborations with developing countries. All right. Facilitating more networking. Absolutely. That is the name of the game here at OCB. That is what we aim to do always, both virtually and in person. Fieldwork opportunities. Absolutely. All right. Thank you. These are fantastic ideas. All right, I'm about to move to the next question, so get your answers in. What is the most important thing OCB can do to support you, wherever you are? My dog has a lot to say about that. Communication, yes. Mentorship, idea incubation, learning opportunities, job and fellowship opportunities. Funding, yes, funding is good. OCB doesn't fund research, but we continue to try to be an incubator for good ideas that can get funded. Continue being awesome. Well, that's a nice thing to say. Whoever said that, thank you. Interdisciplinary spaces, yes. The incubation space, yes. Innovation accelerator is the buzzword here at WHOI these days. All right. Last chance before I stop sharing this. All right. Thank you. Thank you, everybody. So we are at the end. I don't even know who is left here. 29 people. I'm going to do a quick dump into my spinning wheel. Actually, we'll just do it with how it is. And whoever's not, if somebody's not here, they don't get a prize. So there was one person who was not here earlier for the drawing. So that means that I have I believe, 5 more prizes to give away. So the next one is for a utensil kit packet. So here we go. Going to Nicole Poulton at Bigelow. All right, here we go again. Who's our big winner? Peter Hernes. The Arctic Colors folks are making out today. Okay, here we go again. This one's for another utensil kit. Congratulations, Roberta. Okay, is this our last one? No, our next to last one. So this next one is for the OCB double-walled stainless steel water bottle prize. Let's try this again. We go! Congratulations! And the final prize is another utensil kit. Here we go. Oh, you don't get to win twice. All right, let's try again. Let's try again. Raquel, congratulations. Great. All right. That is about all I have. I would like to turn it over to Victoria to formally end the meeting as our incoming SSC chair. And I want to thank everybody and thank you so much for being here. We're going to go to the lobby if you would like for some informal chit-chatting with your colleagues for final farewell happy hour. Thank you, everyone.
Go. I think it would be great for everyone just to kind of give that round of applause to Heather, who's the heart of OCB and has run this meeting absolutely magnificently in collaboration with Mary Zawaski and May Mahegan. So we are really fortunate in the OCB staff that just make everything happen so well. So thanks so much to you guys. And I just want to say, you know, I don't like to fill out surveys at all, but we really, really would like to have your feedback on this difficult and challenging online workshop concept. It's been great to meet so many international colleagues that we might not have seen otherwise. And I want to really just say that, you know, probably a hybrid of online and in-person is the future. So if you can help us understand what aspects work really well and what doesn't, then we can better tailor this meeting to meet what you would like to get out of it for next time. And I think that's a huge service that we can all provide to not just this meeting, but to all these upcoming workshops and more in the upcoming years. So please do think hard about what did you like, what did you not like, how could it be tweaked, and let us know so that we can put in those fantastic plenaries on topics you're really excited about, and continue the posters and networking spaces, or find alternatives that work for you. So I just really, really am grateful to all of you. I hope I got a chance to talk to most of you. If not, I guess there's that happy hour. So I'll just turn it back to Heather and say I hope to see you guys next year.
Thank you, everyone. We'll see you in the lobby.